Hi, everyone. Thank you for joining. Thanks for joining our SCOG meeting. Really happy that so many of you decided to spend Friday morning, at least 45 minutes on learning about some cool new single cell developments. So very dear welcome to our single cell omics network, Germany. Excuse me, a bit of a larger thing. Just added that because you got the great time, but for our SCOG meeting, where Heiko Lickert, as you can see him here, fancy suit, where we're presenting about his research in regeneration research. Before we do that, let me briefly announce a few things before I introduce Heiko. Our next lecture will happen again as our usual cycle in 2 weeks. This will be for Maya from now, just recently ETH, presenting about disentangling tumor unique immune alterations. So essentially using multi-omic analysis in that cancerous context. Quite excited to learn about that. Looking forward to this talk, Florian. In addition, I wanted to mention again a virtual workshop we're doing. Is going to be happening next Monday and Tuesday, where we essentially want to give a little bit hands-on on current tools and the community has been making for spatial transcriptome analysis, in particular in Python. Sort of what we like to focus on, presenting tools from my lab, particularly for Script and Ensign, but tools from Uma Bayraktar and Oli Steger's lab, Cell to Location, and Ekman from Munich lab. And essentially, we have the PhDs and postdocs organizing that thing, and we've been overwhelmed by the reaction from the community. I think we now have more than 550 people joining for that. Initially, we've been trying to do like breakout in smaller rooms. Now we sort of need to change that a bit because of this fantastic people joining that. But if you sort of want to learn a little bit more about these tools, obviously we'll be recording that and sharing that as well, but maybe it'd be fun to join. Thank you. Then last announcement I wanted to say is we're now starting to become a bit more physical again. I don't know how many of you have been going to meetings. I basically said to all my PhDs and postdocs, you have to get out of not only your room, but also the lab and go to one thing, at least maybe 2 things this summer. Just really make use of that. And that's, I think, a general advice. And, if you want to do one of these things, why not come to Munich in mid-September? There we're going to organize a single cell genomics meets data science meeting that will be sort of chaired by Maria Kulomitache from LMU Munich here, Oli Stegle from DKFZ and embryo, and myself. And we really have a whole bunch of fantastic speakers lined up coming in presence to a beautiful location in Munich. Ashley Sanders, Lale, Christoph, Nir Yosef coming, Berkeley, also now Weizmann, Tel Aviv, Stefan Bauer, Magnus Ratreis. So really a whole bunch of sort of hands-on data generation analytics people, but also very data sciencey people, such as Magnus, right? Learning about GPs. And I think sort of bringing this type of crowd together will be quite unique and a lot of fun. Registration will be, as always, first come, first serve. We have something like 200 places available. We're going to be announcing a date, so we'll be somehow end of May. Yeah, please come around. I think that's it, and we should get started. The usual information is we have something like 30-minute talk, then 15 minutes discussion. I will be moderating that. Please raise your hand and we will be unmuting you, or if you can't, for whatever reasons, just put in the chat and I'll be reading up your question. And with that, I think we should get started with Heiko. Maybe Heiko, you can already start sharing your screen while I will be briefly introducing you. So first thing you need to know, Heiko is a legend. So he has been really doing a lot of cool things for a long time and has been in many ways very influential how we here at Helmholtz Munich are looking into single cell variation, as well as a lot of stem cell biology early on, and with a sort of really important transition towards regeneration. Yeah, we've been working together for a long time. Has been one of the key partners we've been doing a lot of stuff with. But not only scientifically, but also sort of managing I think we both had a junior group early on in Helmholtz Munich. So just a short background, Heiko was actually doing a PhD in molecular embryology at Freiburg before then, after some international travels, various places, particularly Mount Sinai, postdoc, starting as a junior group leader in Helmholtz Munich, something like 2005-6, then becoming PI and professor and director of the Institute of Diabetes and Regeneration Research in 2011. Heiko has been having a whole bunch of awards, starting from Otto Hahn for a medal for PhD, Emmy Junior Group ESC starting grant, to I think the most recent, to also an M4 award for company founding and so on. A lot of stuff, sort of really basically translating things, but also most recently, I think most famous one, ESC Advanced grant that he just recently got awarded. And yeah, I just personally want to say that I've been learning so much. I think a lot of my bio knowledge has been basically Heiko pushing me, trying to understand at least some of the molecules, transcripts that he's dealing with, so that we find a joint language. I'm really looking forward to Heiko's talk now about how he deciphers mechanisms of beta cell development and regeneration. Thanks, Heiko, for coming.
Yeah, thanks, Fabian, for the very kind introduction. Yeah, and I have to say, Fabian, I think since we joined Helmholtz, we really found a language between computational scientists and experimental scientists with joint PhDs and postdocs, and that was the way to go, because at around 2005, nobody understood what the other was talking about. So that's key to translate things also interdisciplinary.
We came a long way, right?
Yeah, we came a long way. So so thanks everybody for joining. It's a pleasure to talk here at the SCOG meeting. And today I would like to introduce to you to our favorite topic, which are insulin-producing beta cells in the pancreas. And as you will see in a minute, so they are really important to maintain blood sugar levels. If they don't function, you become diabetic. And it is important to understand where they come from, how they are formed, how they fail, but also that might give you entry points to protect them from stress or even regenerate them. And that is the main topic of my research group at Helmholtz. And we have most of what you will see today is really a great collaboration, long-term cooperation with Fabian, as you heard just now. And without that, a lot of the analysis done would not have been possible. So you may know that 100 years ago, the first patient got actually treated with insulin, and it just took a year till Frederick Banting and Charles Best got the Nobel Prize for that. And already at the lecture, Banting mentioned that insulin is a therapy, but not a cure. And this is now 100 years ago, and still since then, this hasn't changed. So, and that makes diabetes really a socioeconomic burden with ever increasing numbers of diabetic patients. You see, it is over 500 million patients, and the projection line is bad. So the number is steadily increasing because we do not have any causal treatments for the disease. So every treatment we have on the market is symptomatic. It treats the high blood sugar levels. And eventually, the failure of regulating blood sugar levels leads to these devastating secondary complications, which is the burden of the disease, as you can see here. And that is why we really need to find causal treatments. And something to know, what is important is that, and why we're focusing on the beta cells in the pancreas, it is when these cells fail in type 1 and type 2 diabetes, when the pre-diabetic patients actually become diabetic. So failure of beta cells or loss of beta cells indicates progression in disease. And that is then easy to understand for you, that we want to protect or regenerate these cells. We also want to understand how, for instance, autoimmune diabetes really just detects beta cells in the pancreas and destroys them and not the other cells around. What hyperglycemia and hyperlipidemia does to the beta cells in type 2 diabetes. So if you understand the pathomechanisms of disease, you can try to basically protect or regenerate these cells. But currently, we really only have two treatments which can stop disease progression and cure the disease, which are islet transplantation from cadaveric donors. And as all transplantation, the problems here is donor material. There's not enough donors. and you have to deal with immunosuppression after transplantation of these cells. So this is only for a rare number of patients really relevant currently. And bariatric surgery, which is done in morbidly obese diabetic patients. Usually they also have other problems already. But that is a good example that even a week after surgery, beta cells come back in function. So it's a human experiment or surgery model where we can see that even people which are for 10 years on insulin replacement therapy can gain back beta cell function. And this exactly we want to understand how to bring back beta cell function in diabetic patients. Now, and that's why we're working on beta cell protection and regeneration to fight really one of the root causes of diabetes and develop novel therapies. Now, to do so, we have to understand where the beta cells come from. If you want to regenerate cells, you have to understand how they are generated. And during development, as you will see in a minute, these cells are generated from multipotent progenitors. And these multipotent progenitors can give rise to all the epithelial cells in the pancreas, which are ductal cells training enzymes into the gut, acinar cells, which generate enzymes for digestion, and endocrine cells, which find themselves in the islets of Langerhans. And the most prominent here are the alpha cells generating glucagon and the beta cells generating insulin, which are the counterplayers and which regulate blood glucose levels. There is a debate out there if there are any adult stem cells in the pancreas, and I can tell you, because we also study the gut, but if you look in the skin or in the blood, there are really adult stem cells with a high proliferative potential. You don't find these kind of cells in the pancreas. And it is not clear if in large animals and human beings, we have facultative ductal progenitor or stem cells. This is still a question which is out there. During tissue homeostasis, it is really self-replication of beta cells, which adjusts for needs, just like for needs in obesity or pregnancy, where the pancreas has to generate more insulin. So that's when at that time, beta cells proliferate to generate more insulin. So some people try to transdifferentiate close lineage-related cells like the alpha cells into beta cells that requires transcription factor expression. With that, you can convert alpha cells into beta cells. If that becomes clinically relevant to replenish lost beta cell mass, is questionable if you think about how to translate this to human with gene therapy. We are interested in the concept of dedifferentiation. Usually mammals, any tissue, any organ, when it's under stress, loses identity, goes back in developmental history. Unfortunately, we have lost somewhere the regenerative activity which axolotls have, but dedifferentiation is a general mechanism which happens under stress. And if we could redifferentiate these cells, that would be a way of replenishing lost beta cell mass. Now, to understand regeneration, I said we have to understand development. And I will talk about pancreas development now in the first half. So this is the pancreas. It's an exocrine and endocrine gland, as I have said already. There's acinar cells, there's ductal cells, and there is 2% of endocrine cells which produce hormones. During development, pancreatic progenitors give rise to this sophisticated duct-like tree-like organ. And at the tips, you have the acinar cells, which generate the digestive enzymes that get strained into the ducts, into the duodenum for digestion. But then the endocrine cells, they respond to glucose levels and secrete the hormones. And it is essential for us, obviously, to see what signals and factors regulate the early endoderm development, but also pancreas development and then islet development. This usually brings along also the identification of disease genes, because most genome-wide association studies have shown that developmental regulators or mild mutation in non-coding regions contribute to diabetic disease prevalence. And if you have harsher mutations in these developmental regulators, you get actually monogenic forms of diabetes, which are called modi. And so it's also important to understand these building plans and instructions which these cells receive to basically guide pluripotent stem cells into a certain lineage, for instance, into insulin-producing beta cells via every step of development, from endoderm to pancreas to endocrine development. And this, I want to show you how we actually understand development over time to also guide cells into certain lineages. Now, to do so, we have historically generated a lot of knock-in fluorescent reporter lines, because development is a dynamic process, as you know. And you might know from your studies that gastrulation is one of the first processes where embryonic lineage decisions are taking place. The mesoderm, the endoderm, and the ectoderm are separated during gastrulation. And I will talk a little bit about gastrulation in the next couple slides. But then the endoderm gives rise to all the endoderm-derived organs, which are from rostral to caudal, the lung, the liver, the stomach, the pancreas, the gastrointestinal tract. And so to understand how endoderm gets patterned, how organ domains get specified, you have to look at this process in life. And this we have done in mice, and we also now do in stem cell differentiation cultures using fluorescent reporters. And the output here is that you can use these fluorescent knock-in reporters to look at different stages of differentiation in static or live imaging. You can do this in wild type, or you can do this in a mutant context to understand gene function. You can sort populations while they undergo lineage allocation and understand the transcriptional or also the signals they receive. And the output is obviously that we understand early endoderm development, patterning, organogenesis, but also the formation of certain lineages in organs to generate then these cells from stem cells. Now, this is a beautiful example of how you can use reporters. This is from Ingo Burger in the lab. I think this is really one of the most stunning imaging we have done using gastrulation stage embryos, put them under cover slips and image them using confocal microscopy. And so here is the posterior side where the mesoderm and the endoderm forms, where the primitive streak is, and where cells actually leave the epiblast, so the pluripotent progenitor territory, to allocate to the mesoderm and the endoderm lineage. And you can see here that FOXA2, one of our favorite transcription factor, which is also a master regulator of endoderm development, is low expressed in this domain here, in this distal domain of the epiblast. And these are the progenitors of the endoderm progenitor populations. They leave the epithelium and then give rise here. They migrate towards anterior and then give rise to the definitive endoderm, while they intercalate into the visceral endoderm. And so this imaging tells you about where progenitors lie, how the morphogenetic processes go on. And if you then use, for instance, these fluorescent tagged embryos, and here we have used hundreds of these, I think 120 of these or 110 of these early, mid, and late gastrular stage embryos, where we then have sorted the FOXA2 low, these are the progenitors in the epiblast, or the high, which are only a handful of cells per embryo, but sorting them and also comparing them to the negative populations then gives you very sophisticated single cell roadmaps of gastrulation, where you can zoom in very much into these early lineage decisions. And then with Fabian together, we used his sophisticated, wonderful tools like SCVelo or cell rank to basically look at lineage allocations, but also look at lineage driver genes. And so to cut a long story short, that has been published last year, what we have shown is that the endoderm really forms fundamentally different than the mesoderm. So in the textbooks, you can still read that these epiblast cells in the posterior region of the gastrulation stage embryo all undergo epithelial mesenchymal transition. That is a well-established model. But we have shown basically that the endoderm cells never really undergo an epithelial mesenchymal transition, but rather maintain their epithelial identity and give rise to the endoderm. It would not even make sense for them to downregulate a polarity and an adhesion program in the short time when they are leaving the epiplast and contributing to the endoderm layer, because both of these layers are epithelial in nature. And this is not only important to understand basic mechanisms of how a gastrulation stage embryo generates lineages like the mesoderm and the endoderm, but also is essential to understand how cells can disseminate in endoderm-derived tumors. And there are plenty of these. The lung and the pancreas and the gastrointestinal tract are all endoderm-derived organs. And it is important to understand that EMT might not be necessary for metastasis. So after the end of gastrulation, the embryo is really patterned beautifully. It has formed the endoderm. You can see it here marked by two important endoderm transcription factors, FOXA2, which I introduced already, and SCOG17. And you can see already how there is a transcription factor gradient along the anterior-posterior axis. So the embryo has received beautiful pattern information and knows already location, and that is essential also to give the embryo anterior, posterior, left, right, and dorsoventral pattern information, including here with the midline and the node, the organizer tissues. And that can be, the patterning can be even seen more or nicer if you're looking here at a head fold stage embryo. So this is a head fold, so basically this is where the heart forms, but also the head forms, and you have the midline here, and you can see how FOXA2 and SCOG17 forms a beautiful gradient along the anterior-posterior axis. And this transcription factor gradient gets translated into formation of territories which are specified for organ formation. And we know this from old fate maps. So here one from Kim Tremblay and Ken Serrett, but also from Rosa Baddington, Patrick Tam, Kirsty Lawson. So this was where the experiments done in the 80s, 90s, where cells were labeled and then looked what fate do they allocate to. And from these fate maps, we know that already at this stage, organ territories are formed and specified via signaling gradients, but also via transcription factor gradients. And the question for us was, can we actually translate this also to the culture dish where you basically do not have organizer structures? Because we know a common problem in stem cell differentiation is that it's intrinsically heterogeneous, and it starts already at pluripotent state. Cells are heterogeneous throughout differentiations. And how do you guide them into the right direction? For instance, liver versus pancreas. And so here we have actually shown in a very nice paper from Kati and Pahlavi that we can identify subpopulations of endoderm using surface markers, which are lineage biased towards the liver and the pancreas. And that helped us to understand also the signals, which are essential for this lineage allocation. But more important is we could sort pancreatic progenitors and could show when you have more homogeneous differentiations that you can get more functional cells. In this case, more functional glucose responsive, insulin secreting beta cells. So this shows you that understanding early development is really essential to guide cells in culture dishes to functional cells. So we need this embryonic developmental information. So moving on to the pancreas. So this is now a beautiful imaging also from Ingo, pancreatic explant cultures. So we take a pancreas here at embryonic day 12.5. It's marked for FOXA2 venous, PDX, BFP. And what you can see here is the acinar cells. These are the cells which give rise to the enzyme producing cells, the ductal cells here, these cells, from which these are the progenitors during development, and from which the endocrine cells, then these are the orange cells which wander around. These are the cells which leave the epithelium and find together in the islets of Langerhans. And you see this double reporter is fantastic to look at basically pancreatic explant cultures. You can understand morphogenesis here. You can understand how the organs form. You can perturb the system, but you can also use the system or other reporters. For instance, here we have used even another reporter, which is the NGN-free venous reporter. And neurogenin-3 is the master regulator of endocrinogenesis, and is only very shortly expressed in these ductal progenitors. So basically in the cells here, which are orange and leave the ductal epithelium, and then NGN-3 expression is gone. So this is a very short time window. And so having this reporter allowed us to basically enrich for the cells we're interested in the endocrine progenitors, to understand how endocrine cells are formed and how alpha and beta cells are generated. So we used this reporter and did single cell RNA-seq. So this was, again, a fantastic cooperation between our experimentalist and also the computational biologist. I think this was also a role model for Fabian's group to generate several algorithms and several new tools. For instance, the SCVelo, but also the cell rank tool was trained or established on these data sets. And for us, it was great because we got the finest fate mapping and resolution of how ductal progenitors go via endocrine progenitors towards alpha, beta, epsilon, and PP fate. But now the important thing is that basically all what we know is based on mice. Why? Because we can manipulate mice, but mice are not men. And we would like to generate, obviously, from human stem cells, cells for cell replacement therapy or for disease modeling. And that is why we have to understand also human development. And that is lacking behind very clear, because there is no knockout experiments you can do. It's very hard to get basically embryos, as you can imagine, and to study embryonic development in human. But there are fundamental differences. I mean, the most obvious difference is that basically the mouse develops in 3 weeks and human beings develop in 40 weeks. And there's even tissue interactions which happen in mice. For instance, here the notochord is juxtaposed to the foregut, and there's important hedgehog signals coming from there instructing this region for pancreas development. But in human beings, the notochord is not even juxtaposed to the foregut because the dorsal aorta is already generated. So the neighboring tissue interactions are not even taking place in the same manner in these different species. There's differences with the transcription factors when they turn on, which transcription factors are used, which signals are used. And these differences, if you think about that, all the differentiation protocols are generated based on knowledge we have from mouse, already tells you that we would, if we would understand human development better, we could probably bridge this gap, the translational gap or evolutionary gap, a bit better to generate better differentiation protocols. So we really have to understand large animal development. And because human is difficult to access, we have adopted the pig. And for a simple reason, we have Eckhard Wolff, Angelika Schnieke here in Munich, which can generate transgenic pigs. Eckhard Wolff has a lot of diabetes pig models. And the pig is actually also a model for xenotransplantation. You have heard the news that a pig heart was transplanted into human. Our Dresden colleagues, Barbara Ludwig and Stefan Bornstein, they are trying to get xenotransplantation for islets into the clinic as an alternative cell source. And there's many reasons why you should understand pig more in detail. For instance, pig insulin was used until recombinant human insulin got onto the markets in the 80s. So until then, pig insulin was used for insulin replacement therapy in diabetic patients because it only has one amino acid change. Pig metabolism is very close to human, even closer than non-human primates in terms of glucose control and C-peptide secretion, but also islet of Langerhans structure. But surprisingly, there's nothing really known about pig pancreas development. Nobody really bothered to study this. There's maybe a handful of papers out there. And that's why we wanted to understand how the pig pancreas forms. And so here with Eckhard Wolf together, Fabian Theis, we then assembled a beautiful single cell data set over the first, the second, and the third trimester of development. The third trimester is not shown here. So pig development goes to day 110, so much longer than mouse development, more closer to human. And so here we sampled cells over time, and then we generated a pig embryonic atlas. And when you're looking at this over time, it's just beautiful how you can see the differentiation waves going through the tissues from multipotent progenitors, which allocate to pre-acinar cells, pre-ductal cells. But then also there is a paucity, there's two waves of NGN-free cells forming endocrine cells, an early and a late wave. And there's a paucity in between. But from day 40 onwards, you can see that most the bulk of the endocrine cells in the second transition is formed. So this is closer to mouse. Human only has one endocrine transition, one wave. The interesting thing about having this development stretched out is that you can actually look much better at lineage transitions. You can resolve that much better over time, because what you see here is actually compressed in the mouse in a few days. So in 3 days, this happened. But here we're looking at weeks of development, and we can look at lineage bifurcation in much higher resolution, in much higher temporal resolution, but also molecular resolution. And we can identify the lineage driver genes, which are essential for alpha cell and beta cell lineage allocation, which basically is not yet really understood. So the differentiation protocols, there is alpha cell protocols out there, but most is geared towards beta cells, and the upstream signals are still not yet understood. So because NGN-free is so important, we then also generated a knock-in pig together with Eckhard Wolf, and the strategy worked beautifully. So we used an NGN-free venous. So this shows you the transient expression of these cells in the ductal epithelium, and then used an H2B scarlet as the lineage tracer. So we generated a chrono reporter where we can actually fine resolve the lineage allocation even better. And that is good to understand how endocrine cells form during development, but it also will help us to understand how the pancreas undergoes the tissue homeostasis in the adult states. So if there are endocrine cells formed by neogenesis from ductal progenitors, so this one question which is still outstanding and which is not solved in large animals, if there are ductal progenitors giving rise to endocrine cells when there is need, for instance, in pregnancy or in diabetes progression. Fabian?
Just a few minutes, wanted to say.
Alles klar. Wie viel? 5?
Yeah.
2? I'm almost done. All right. Thanks. So, and wrapping up here, this developmental part is, it's really like the developing pig pancreas has taught us that multipotent progenitors are actually maintained very long during the first trimester. We can look at how multipotent, bipotent, and unipotent lineages arise, and we learn much better about endocrine lineage allocation. And so what we're doing now is we are comparing via also multi-ohm single-cell roadmaps, mouse, pig, and human development to look at species-specific, but also to look at conserved mechanisms of development. And that is essential to generate new differentiation protocols, but also to basically understand how we can translate things which we find in the mouse towards human. Now, as we're running a bit out of time, so yeah, this is the evolutionary comparison is really essential for translation. This is what I said. And we add a new model system, which is the pig, to this comparison. Because we're running a bit late, I would actually say I would wrap up here and skip the little regeneration part, because it's shorter. And I think maybe it's better to have a discussion here on the developmental part, which is so essential for regeneration. So I would go and, let me see, go to the last slide, basically, which is here. So important, most important is to thank all the people which have made this work possible, and also the funding agencies. But we have great people in the stem cell area. We study pancreas development, as you have seen. I have not talked about insulin signaling, which we're lately interested. We look at islet biology in the adult, and we need great cooperations, like with Fabian's group. I think there's not all of the Thais group listed here, only the ones where we have published yet. And we have other great cooperation and great funding. So I would stop here and would be happy to take some questions. Thanks for listening.
Awesome. Thanks, Heiko. This is really cool. Thanks for taking the time and sort of having this tour de force across so many different things. And I've been always trying to puzzle in my head how to sort of add the peak variation on top of that. And I kind of get that this is sort of between human and mouse, but I think there's a whole bunch of cool things that can be done there. And maybe mouse that doesn't see. We have time for questions, and I think Heine has really one. Do you want to unmute yourself and ask?
Okay. Hi, Heiko. Haven't seen you for a while.
Heine, nice to see you.
Good, Steve. Okay, so question about endoderm formation. So I remember our old discussions when you always encouraged me not to talk about mesoderm formation and endoderm, but always on mesendoderm. So, but now it looks that there is really a specific mesoderm induction and endoderm induction. Do you know the signals, what really separate these two?
Yeah.
And is there also Wnt signaling inactive because there is no proper EMT?
Yeah, exactly, Heiner, that's the mechanism. So we generated a procurie, which you know very well, a T reporter, and a FOXA2, so a double reporter. And what you see, I think this is already published in the 2009 Ingo Burger's paper, yeah, that already in the epiblast, you can see that when there's just a few cells induced to become mesoderm or endoderm, and I would actually not talk about mesendoderm anymore. It's confusing, yeah.
Yes.
Because they already segregate in the epiblast, yeah, and you can see that the mesoderm, the T positive cells, yeah, they are more distal, more proximal, and the endoderm cells are more distal, and you can see this even the primitive streak, the anatomical position of the primitive streak, the endoderm cells are always anterior to this. Versus in the primitive streak, you have several cell layers, you know that much better than I am. So there's 2 free cell layers in the primitive streak, yeah, and these cells, they have received Wnt signal, yeah, TJ beta signal, they undergo the EMT, they become mesenchymal, do the classical ECUT-NCUT switch, upregulate EMT transcription factors, lose polarity, yeah, and become with vimentin, and so become mesenchymal. Versus the endoderm cells, they leave the epithelium, we think they just shortly downregulate on the protein level, caterians and polarity molecules, they get squeezed out in front of the streak, and then there's just one cell layer in between the visceral endoderm and the epiblast. And they basically, because of ECUT-NCUT double positive expression, yeah, they get sorted out and get incorporated into the visceral endoderm, and there's no EMT there.
Yeah. Okay, and may I ask a second question, very short, briefly? So, is there no induction of a dorsal pancreas in human, as in mice? Because you said that the dorsal aorta is separating notochord and foregut.
No, there is dorsal and ventral pancreas, yeah, but the signal
without activating signal.
the hedgehog from the notochord, yeah. The hedgehog from the notochord, I think they experience it only for a short while, very short, yeah. But then the dorsal aorta squeezes in between, yeah, And then that signal then disappears. But you have a dorsal and a ventral bud. That's the same, yeah? And also the dorsal and ventral progenitors will give rise. So we know that to the head and the tail of the pancreas. And there even the endocrine cell compositions are distinct, yeah? And I think it's not proven yet. Nobody has proven this, but this developmental origin probably makes this difference. We have to prove this by fate mapping, yeah?
Okay, thanks, Heiko. And hopefully I will see
good to see you, Heiko.
Okay,
see you
take
soon.
care. Thank you.
Maybe Heiko, we can stop sharing screen so that we can see
yeah,
people sort of showing themselves. Should really happen. A brief question from my side, what people hopefully think about cool things. There's been so many atlases coming out recently, right? We have these embryo development atlases, in particular also parts driven by Antonio Scaldone here at Helmholtz Munich, and of course many others. There's been sometimes people adding time resolution, sometimes people adding ATAC to maybe understand more regulation or spatial context. I guess it might be fun to integrate, but let's say we had all of this in super high resolution. Let's say we had like this embryo development atlas fully done with all the modalities. What would be questions that you sort of could then leverage this for? Because I think in some aspect, we're very much working on just doing the counting and the resource building, but I have the impression only, you being very much closer to sort of deeper questions would be sort of leveraging that. So what would you use that for? Would you like just project your data on top of that, or what would be sort of things that would be?
Yeah. No, I think, one important thing is human cell atlas, yeah? If you think about it, okay? So how do you get the cells actually? And it's usually, if you want cells from an adult being, yeah, it's post-mortem tissue. And it is, it's a snapshot. It's one time point, yeah? Embryogenesis in human is not available or not accessible, yeah, beyond the second trimester, yeah. So you just think about this. So I think what, for instance, also with transfer learning or what you guys do, what I could imagine is, if you have, for instance, you have a mouse, you have a pig, yeah, where you can actually access all, let's say, tissues with the best quality you can imagine. Human tissues are usually also not great quality, as you probably know, yeah, if you have to wait for post-mortem tissue, yeah, shipped around the world. Yeah, so you could have nice maps of model organisms across evolution, and then have, let's say, what is accessible in human, but try then with transfer learning, for instance, also get some stages which are not accessible, yeah, or what, I make you another example. If you want to have a type 1, early type 1 diabetes pancreas, you have to screen 5,000 organs. Can you imagine? So this is super rare. But if you want to understand disease progression, you will need to stage, right? You will need to go from healthy pre-diabetes to diabetes. Yeah. How do you get this projection line? Yeah, you need this tissue. And I think there the model organisms are helpful. And I think the human cell atlas has also model organisms in there, right? And they need it. And I think that's—
I see the point you make it is we should now add like progression type of time series or something like that, or maybe not time series, maybe just like progression stages on top of it, and then connect. I mean, maybe to the others who might not have seen that, I thought that was really one of the most fascinating sort of analysis we've been doing, which we initially didn't actually expect. Because usually we do pseudo time within a developmental organ or something like that, right?
Exactly.
Or sometimes maybe spatial localization or relocation, but usually that. But in Heiko's case, the idea was that you had, I think, this de-differentiation of beta cells across stages. And obviously these were very different animals. I mean, this was different disease stages, but you could also see sort of similarities in that transfer space. So you would argue that this similarity, like this old Waddington's landscape type of idea, would still hold there and would sort of see how it's being rewired in a disease?
Yeah, probably yes. I think if you take the single cell omics approach, yeah, I think where do you get the nicest data? Yeah, it's usually the systems which are dynamic. But now in an adult human being, for instance, there are a lot of the organs have formed. Yeah, the dynamics are there's no more dynamics. It's a steady state. But then when you have stress, then it will get dynamic again, because there is obviously defense mechanisms you want to understand. There's some, even this question is not yet clear with dedifferentiation if it happens in human, but it will come through systematic profiling of different stages and looking at that. And I think then when we need a time series. How do you get the time series? Usually it's endpoint analysis, and we cannot even define what is a healthy human being. Please define healthy normal. So therefore I think you need a staging, you need time. It's good that we can do single cell, but we need the time always. It's like the example with the reporters we use for embryonic development. If you want to understand development, you have to understand how you get from A to B without a black box in between. And I think there the AI thing can obviously help us. If we are not getting all of that, and it will be, accessibility of tissues is difficult. But there I think AI can maybe bridge some gaps and make some predictions. That's how I see it currently. And obviously,
I get this layer,
Fabian, every extra layer, if it's now, let's say, chromatin opening, or if it's RNA to protein, or where does it happen? I mean, where does it happen, for instance, is for us when we have the reporters, we know already. So
in your case, spatial would not be even adding up as much as we're saying.
Not too much, probably.
Thanks so much.
Look at a chrono reporter, for instance, right, where you already have a fine resolution.
I mean, I guess it's kind of hard in pancreas anyway, right? Sorry, I should not occupy that discussion. Let's just take one more question, maybe not so we're getting close to the end. There was Caesar asking about whether you have seen the C-cell phenotypes or gene programs similar to the development ones. And Caesar, if you want to unmute, of course, you can sort of specify that. It's fine.
But I think it's fine, I mean, it's a very important, that would have been the second part, which I skipped, yes, so we basically make, for instance, animals diabetic, and then, understand what, for instance, with these islet cells happen under diabetogenic stress, how do they react to them? And do they go back in developmental history? And I think they go back in developmental history, but in our field, there's quite a debate out there how far they go back, because there was one initial paper which was suggesting that pluripotency marker come up. Maybe if you're more familiar with regeneration, yes, the axolotl is doing this. If you cut off the limb, then the blastoderm will go back in history to a pluripotent state, and then can form all the cell types again to regenerate. We have lost this in evolution, but still I think that cells under stress, they go back in developmental history to a certain point, not to the point of pluripotency, but rather to a point of an endocrine progenitor. And then from there, we need to understand how we can bring them back. And that requires understanding of, embryonic development is not finished until birth, right? Because then, for instance, the lung needs air, and that is a maturation trigger for these cells. So we need to understand these these developmental processes which happen after birth, maturation processes, because that is usually from having an identity to become functional. That little step, I think that's where when stress comes, that gets lost, and then cells are unfunctional. And that is what you would like to prevent, or that is what you would like to reinstall. And this we have shown in a nice paper from Stefan Sachs, also together with Fabian Theis, as an example for islets under diabetogenic stress, and what happens if you do pharmacological treatment, how you get them back on this developmental trajectory towards a normal state. I hope this answers your question.
That's fine. So thanks again, Heiko, for this really great talk, and very nice to see where things are going. Heiner, did you want to follow up on that? I see you muted. Heiner, you're muted. Heiner, we can't hear you. You're muted.
Oh, but both is on. You hear me now?
Now, perfect.
Yeah, okay. So transdifferentiation between liver and pancreas was really a long discussion in the 20 years ago. What is actually happening on this field?
I have the feeling it dies a little bit out because it's too wild. So, but what the people look at, Heiner, is a little bit more into in the gut. You know, the gut is the largest endocrine system and has a lot of endocrine cells. And people are trying now, because they have seen when they knock out certain transcription factors, yeah, that they can actually get insulin-producing cells in the gut. Yeah. So people focus on this, and then other people focus, what I said before, like Pedro Herrera focuses on alpha to beta cell transdifferentiation, because the lineages are closer related. And in type 1, the beta cells get killed, and then you have too many alpha cells. Yeah. So the idea is interesting. Yeah. If you then just convert the alpha into beta, then you establish the steady state again. But then you have to think about how do you deliver this in a diabetic patient. Yeah. So it's a bit difficult, right? I mean, are you doing gene therapy if there's metformin or insulin out there? I'm a bit skeptical that will happen.
Okay, thanks. Bye.
Yeah.
All right. Thanks again for the fantastic presentation. It was really exciting to discuss. Thanks for coming. See everyone in 2 weeks, maybe next week ready for the workshops, and yeah, catch up later physically, September Munich. See you around. Bye. Thanks, Heiko.
Bye.